Okay, this is the second of the preps for lecture 15. And where we had finished, I had just I went pretty slowly through the actual cross, and I rewrote it a few different ways to try to take us through the reasoning that brings us to this, um, with the genotypes written this way, which is correct notation for genes that are linked. And I finished off with just the calculation of the percentage of recombinants. And we'll talk about this later when we talk about what we would use that calculation for, and we do some practice calculations together. And I had asked you to just imagine that the cross had been done a different way. Suppose one of the parents had wild type for the eye color gene, but mutant for the wings, and then crossed to mutant for the eye color, but wild type for the wings. The dihybrid would still be wild type because it would still be heterozygous for the two genes. And we would still do a test cross. And we would expect to see the same kinds of results. And we do. The numbers aren't exactly the same, but they're very comparable. Here we have, and these are the real results. So you see they don't add up to like exactly 2,000 or something like that. They are, actual numbers. But there are two large categories of progeny. One that's wild type for the eye color but mutant for the wings, and one that has the mutant eye color but the normal wings. And then there are two recombinant classes. They're seen much less frequently, and they are wild type for both or mutant for both. Okay, and hopefully the color coding, I'm not going to rewrite it as many times, but I just included the color coding just to make the point. And the thing to really pay attention to is that the two categories that are the most frequent categories are the ones that have the same phenotypes as each of the original parents. So this one is like this parent, and this one is like this parent, right? So in both cases, even though the actual genotypes that we see more frequently are different, if you go back and look at each of the two crosses, these are the ones that are most frequent, the ones that we saw in the original parents. And that is one of the reasons why we call, these are the non-recombinant classes or the parental classes, and these are called the recombinant classes or the non-parental classes. And so those terms are used pretty much interchangeably in this kind of stuff. Okay, and so if we rewrite it, given what we know about the relationship between those genes, then they're written this way, right? This shows us, and this is a trans dihybrid, which I'll talk about in a minute. Okay, so there's two different possible arrangements that we can see with dihybrids for linked genes. And in one case, the dihybrid has from one of the parents a homologue that has the two dominant alleles on it. And the other homologue has the two recessive alleles on it. And this is called cis. Cis means beside, actually. So I believe, too, I recently read or heard that it's Greek and it should be pronounced kiss, but everyone says cis, and I don't think we're ever going to change the pronunciation of that. It would also be a confusing term if that was the case. But in any case, we call this a cis dihybrid, two dominant alleles together and two recessives. This is a trans dihybrid. Trans means across. And so in this case, across because one homologue has one of the dominants, but the other dominant is on the other homologue, right? And so this may not sound intuitive, even if you know what those terms refer to. So you might have to just memorize. But once you have, then if you remember that, it'll be helpful. Now, they used to sometimes be called coupling and repulsion, whereas with coupling, it's the two dominants together as a couple, sort of, and the two recessives together as, you know, two coupled. Whereas repulsion, it's sort of it's as if they're pushing away from each other. I don't find this actually very intuitive. I think the cis and trans makes more sense to me. And if you've taken chem courses, you'll hear about, you know, cis arrangements of alleles and trans fats and things like that. So that might come more easily. Whichever term you find more intuitive and easiest to remember is the one you should use. Okay, so now if these genes are linked together, if they're connected on the same chromosome, how is it that you can get the non-parental combinations at all? If you took 102 last fall, you might think it was impossible to get the other combinations, but it actually isn't. And in fact, it's a very useful thing. We can use it to find out how far genes are apart on a chromosome. And there's way more to how crossover occurs, but for the purposes of this class, we're not going into the details of that. I believe in some of the MBB courses, you may learn a little bit more details of, you know, at the molecular level. But all you need to know is that it's a kind of breakage that occurs, and the two pieces are swapped, so that after the crossover event is over, you still have your replicated chromosomes, but this is no longer completely, say, a maternal chromosome, or this a paternal chromosome, because one of the chromatids is still intact exactly as it was inherited from the parent, but these ones have been rearranged, right? This has an allele that was originally on this chromosome, and this one has an allele that was originally on that chromosome. And it's called crossover because when you physically look under a microscope, you can actually see kind of an X-shaped structure, which is why we draw it that way. You can see the actual place where that crossing over is taking place. And this is a schematic, but I have a micrograph too, an electron micrograph. And so in eukaryotes, when we have the pairing in meiosis 1, only in meiosis 1, where one homologous chromosome that's replicated is paired with another. The reason why they pair with each other is because their sequence is almost the same all the way along the chromosome. And these breakage and rejoining events can happen along the chromosome. And you can physically see the structures. And in this case, the crossover is occurring between chromatids 2 and 3 here. So 2 on this chromosome and 3 on this one. This is another possibility. This one is between chromatid 3 here and chromatid 1, and so forth. Okay, and you can physically see them. And so you might want to think about, we never see these kind of structures in mitosis, and you just imagine why, and that will help you confirm that you understand how meiosis and mitosis are different. Okay, and this is an actual electron micrograph that was taken that shows these two structures. These are the centromeres. These are our chromatids. These are our homologs of each other, and you can see the place where there is something going on, and when that's resolved, you know, this part of this chromosome is going to be attached to this, and vice versa. So we actually see those things exchanging. Same down here. There's an exchange taking place. Now, if you looked along the length of a chromosome, at every position along the chromosome, there's an equal probability of crossing over. So it doesn't happen only in one place. There are certain systems that have site-specific crossing over. It's a very different thing, which you don't need to know about at this point. So when we look along the length of the chromosome, for the most part, there's an equal chance that the crossover could take place here, or here, or here, or here, or here, or here, with the corresponding part of the other chromatid. So you can imagine that the more distance there is between two positions, the more likely it is that crossover can occur, right? If two genes are like this far apart, the chance of a crossover occurring right exactly there is very small. But if you've got all this space between I should really draw them sort of like this. If you've got this much space between this position and this position, there's lots of places where a crossover could take place, right? And we'll come back to that in a minute. Okay, so this just reminds you of that sort of that meiotic input and the output kind of thing, and just pointing out that recombination, as I introduced previously, it's the production of these new allele combinations that are not the same as the P generation, right? And that they can arise two different ways. One is independent assortment, and if the genes are not on the same chromosome, the recombinants are produced by independent assortment. The recombination frequency is going to be 50%. It can never be more than that. And if you're not sure why, you should kind of draw it out and remind yourself of how that works. And then if the genes are linked, the parentals are always going to be in more frequency than the recombinants. And so we're going to see fewer than 50% of the non-parental types, right? Yeah, and so this is one of the first things we look at when we do a test cross or something like that, is we go, okay, well, how many non-parentals do we see? And if it's like 48 or 49%, oh, we don't really know. It could be unlinked genes and just a little bit of variation in the numbers, or it could be genes that are quite far apart but on the same chromosome. But if we see 10% recombination, like we did with purple and vestigial, 10 or 11%, then that's the evidence we use to show that the genes are on the same chromosome. And the further apart two genes are, the more likely it is that there is going to be recombination. So what we can do is we can use how much crossover we can detect when we do crosses as a measure, a direct measure of the actual physical distance between two genes on a particular chromosome. Because this just shows that when genes are closer together, there's fewer chances for crossing over to occur between them. But when genes are further apart, you have multiple recombinants. You're going to get a higher frequency of crossing over that gives us non-parental combinations. And this is not just, a lot of, I think, students feel like this is some sort of just exercise, right? But it's a real thing. When we do the calculation, and geneticists do these kinds, not so much anymore, but we used to do these a lot more than we do. And they actually meant something very real, which is the actual physical position of a gene on a chromosome relative to another gene. Okay. And so the way we do it is we use that frequency of crossover. So we calculate a value of R. R means our recombination frequency. And that is, we just total up the recombinants, and then we divide that by the total progeny. And we can only do it this way if we do a test cross. It has to be a case where every recombinant can be easily identified based on its phenotype. And if you want more information on that, you can ask me, but just otherwise remember it has to be done with a test cross. So we take all the recombinants like we did with the purple versus vestigial, divide by the total, and we get whatever that was, 10.8 or 10.9%. And so R in that case would have been R equals 0.10, whatever it was. I can't remember. Let's say it was 8. But then for mapping, we want to convert to map units. So we convert it into a percentage. We multiply it by 100, 10.8% or 9 or whatever it was. Okay, and we have 1% of recombination crossing over is 1 map unit. And again, as I said, mapping is a real thing, and a map unit, for most organisms, there may be some exceptions, but for most organisms, a map unit is close to like around 100,000 base pairs, right? So if we had 2 genes and we were getting only about 1% crossover between them, we could guess that there are about 100 to, maybe between 90 and 120,000 nucleotides apart. And that's helpful information if you're trying to figure out what gene is responsible for a condition and you know approximately where it is on a chromosome relative to a gene whose position you know, right? If it's 2 map units from that gene that you already know where it's located, you're looking a couple hundred nucleotides away from that to find the gene that is responsible for your condition. And this is referred to as recombination mapping. Okay, so the position of a gene on a chromosome is called the locus. This is the pronunciation because it's not obvious how you pronounce this. So that's the singular, so one locus and multiple loci, okay? We take a lot of these terms that we use from either the Greek or the Latin, and so sometimes the pronunciation is not super obvious. So I try to always throw that in when I can. And so whenever we find that the locus, the position of 2 genes on the same chromosome are close together, then we say that they're linked. If they're completely linked, so close that there's no crossover, then the R that we calculate is going to be 0. This partly depends on how many progeny we look at. I was once involved in supervising some undergrads who were working on mapping alleles inside of a gene, and they were collecting recombinants, crossovers from one part of a gene to another part of an actual gene. And they had to look at, I think it was 20,000 progeny to find a few crossovers. So we would call genes completely linked if we weren't able to detect crossovers between them for the purposes of the experiment. But in physical reality, there's always some nucleotides between the genes. And if the 2 genes are unlinked, that is segregating independently, R is going to be 0.5. So this is something for you to keep in mind and to keep in your mind because the value of R is going to be either greater than or equal to 0. It can't be any lower than 0. You can't have a negative R value. And also it can't be any higher than 0.5. And if you want an explanation for that, you can take my 302 class, but I can also talk to you about it if that's something that you're interested in. We can just do a few calculations. It would be better to wait until after lecture 16 before asking that question. And I think I will stop because I'm going to go through now a few examples and leave you with a question or two to answer. And so I think it would be better to give you some time in between.